Welcome back all of you to the online class of biology. We were studying chapter 8, Cell, the unit of life. So in the previous videos we have studied about various organelles that are part of endomembrane system, and we have also studied the structure of mitochondria, chloroplast, their functions, etc. So today we are going to study, and also we have studied about the ribosomes. So today we are going to study about the cytoskeleton. So cytoskeleton consists of filament-like, proteinaceous structures that are present in the cytoplasm, is called what? Cytoskeleton. So what is cytoskeleton? They are network of filamentous proteinaceous structures which are found in the cytoplasm. And the major function of them are mechanical. To give the mechanical support. It gives strength to the cytoplasm, and they help in motility. They help in the maintaining of the shape of the cell, etc. So cytoskeleton is structurally what? Proteins, and they are filamentous structures present in the cytoplasm. And the major functions of cytoskeleton are: they help to give mechanical support, they help in motility, they help in maintaining the shape of the cell. And the next structure that we are going to study is cilia and flagella. So cilia and flagella, they are helping in the locomotion. Understood? So either the cell can move with the help of the cilia or flagella, or they help in the movement of the surrounding fluid. They just work as the oars of boats, with which the boat can move. When the oar is moved, then the boat will be moved, isn't it? So like this, this cilia are helping the cell to move, or they help the surrounding fluid to move in one direction. Then cilia and flagella, when we compare, cilia are small structures, but flagella, they are longer and responsible for the cell movement. Then you can see flagella even in prokaryotic cells like bacteria, but their structure, if you see, that will be different from the eukaryotic flagella. So let's study in detail about the structure, how it looks, what are the components of a cilia or flagella. Cilium is the singular of cilia, and flagellum is the singular of flagella. So when you see the structure of a cilia or flagella, you can see that it is covered by a plasma membrane. Plasma membrane will be there, and the central core is known as axonym. What it is? Axonym. Understood? And you can see that axonym. And in the axonym, you can see a number of microtubules arranged parallel to each other. Parallelly arranged microtubules you can see. And this axonym is having 9 pairs of doublets of radially arranged peripheral microtubules. I will show in this diagram. That is, this is the axonym. And in the axonym you can see microtubules. They are proteinaceous rod-like structure only, and they are arranged parallel to each other. And peripherally you can see how many? 9 doublets. Means 2 are there here. 1, 2, 3, 4, 5, 6, 7, 8, 9. So in this way 9 doublets. Doublets means 2. They are arranged in 2. 2 doublet or doublets of 9 microtubules are arranged peripherally. Okay, and in the center there will be 2 microtubules. Okay, and you can see that this arrangement of microtubule is referred to as 9 plus 2 arrangement. Okay, so this is the typical structure of a eukaryotic eukaryotic filament or cilia. Okay, 9 plus 2 arrangement, and you can see that the central tubules. Okay, the central tubules are they are connected by a bridge. Okay, you can see here some structure is there that connects this two, and there will be a sheath that connects the the two central microtubules. Sorry, there will be a bridge that connects these two central microtubules, and there will be a sheath that covers the central tubules. Okay, then you can see that the doublets, from each doublet, one of the microtubule is connected to the central sheath by certain linkers. Okay, so this is the linker, okay, with which one of the doublet, microtubule of the doublet connected to the sheath. Okay, central sheath, that is known as what? Linkers. Sorry, they are not the linkers, they are the radial spokes. Okay, so this is the radial spokes. Which one? This one is the radial spoke. What is radial spoke? Radial spoke is the structure with which the microtubule of each doublet of the peripheral region is connected to the central sheath, and this adjacent doublets, they are also connected to each other by linkers. So this is the structure of a cilia or flagella. Okay, so if the question is asked, explain the 9 plus 2 arrangement of cilia. So what you have to write? You have to write that the cilia or flagella will be covered by a plasma membrane, and the central core is known as axoneme, and in the axoneme you can see microtubules arranged parallel to each other. Okay, and this is a cross section, isn't it? This diagram is showing the cross section. Understood? And you can see that there are 9 doublets of microtubules arranged peripherally, and 2 of them are arranged in the center. Okay, and the central microtubules are they are connected by a bridge, and it is also covered partially by what? A sheath. Central sheath is there, and from each doublet of the peripheral microtubules, one will be connected to the central sheath by radial spokes. Okay, and the adjacent doublets, they are connected to each other by linkers. Next is the centrosome. Okay, centrosome is an organelle having 2 cylindrical structures called centrioles. So centrosome is the organelle having what? 2 centrioles, and they are arranged perpendicular to each other. Understood? So if you see the structure of this, that is almost same like the structure of the flagella, but there are certain difference. That is that is made up of 9 peripheral fibrils of tubulin. Tubulin is one kind of protein only. Okay, so you can see that it is made up of 9 fibrils of tubulin, and each of the fibril is a triplet, but here in the flagella it was doublet, isn't it? So here 9 what you can see peripheral fibril triplets you can see, or fibrils triplet you can see, and each of them are, each triplet is also linked just like the the flagella structure, and the central part is proteinaceous and is known as the hub. Okay, and which is the hub is connected with the tubules of the peripheral triplets by radial spokes, just like the what the flagella. Okay, so the centrioles, they form the basal body, or the cilia or flagella originates from the centrioles. Okay, and spindle fibers, have you heard about spindle fibers? These are the structures which appears just before the cell division. Okay, and during cell division, especially in animal cells, okay, the spindle fibers originate from the centrosome, okay, and they get attached to the chromosome. So that we will study in the next chapter when we study the cell division. Understood? And they help in the cell division, the spindle fibers. So spindle fibers, cilia, flagella, etc. are originating from the centriole. Okay, so is it clear? This is almost same, means the structure is almost same like what the structure of flagella or cilia. Okay, means for study purpose it will be easy if you compare and study. Okay, there will be peripheral fibrils made up of triplets, okay, of what tubulin, and then the central part is known as the hub, and each of the triplet will be attached to the hub by radial spokes, and these triplets, adjacent triplets, are also linked. Clear? And what is the function of centrosomes or centrioles? Centrioles forms the basal body of cilia and flagella, and they help, okay, in the formation of spindle fibers during cell division. So hope this much is clear to you. Next topic that we have to study is nucleus. Okay. So as you know, the nucleus was first discovered by Robert Brown, and later on the other material, that is some what material which is stained by basic dyes, was given the name chromatin by Fleming. Okay, so the material of the nucleus, so that was stained by basic dyes. When we stain the things, okay, you have done the staining procedure, isn't it? So likewise for nucleus, okay, when it is stained by basic dyes, in the nucleoplasm some materials were getting stained, and these were called as chromatin by Fleming. Okay, so when a cell, when a nucleus is observed in its interphase stage, what is interphase? That also we will study in detail in the next chapter. Interphase is the stage where the cell is not dividing. Okay, so the nucleus of an interphase cell, if we observe, you can see that, okay, there will be a membrane known as nuclear membrane, then there will be a matrix known as nucleoplasm, and in that you can see highly extended and elaborate, okay, means entangled mass of nucleoprotein called chromatin. Okay, so you can see that the nuclear membrane, okay, so what all you can see? Nucleoplasm, then nuclear membrane, then nucleoplasm, okay, and in the nucleoplasm you can see chromatin fibers, okay, or chromatin material, and you can also see some spherical bodies called nucleoli, or nucleolus is the singular of that. Understood? Then if you see the nuclear membrane, they are having two different layers, the outer layer and the inner layer, and in between this there will be a space. That space is known as perinuclear space. What it is? Perinuclear space. Okay, perinuclear space. And then, and the matrix, okay, and I told the membrane is having two layers, outer and inner layer, okay, and in between the space in between the two membrane is called what? Perinuclear space, and the outer membrane is in continuation with the endoplasmic reticulum. We have studied that, isn't it? And you can see ribosomes attached to that. Okay, the outer membrane is continuous with the endoplasmic reticulum, and you can see that there are pores, there are certain gap within the nuclear membrane. They are known as nuclear pores, and this nuclear pores are very important for the passage of RNA and protein molecules in both the direction, that is from nucleus to the cytoplasm and from cytoplasm to the nucleus. Okay, various molecule has to be exchanged, and this takes place through the nuclear pores. Okay, and usually there will be only one nucleus per cell, but there are examples of cells having more than one nucleus. We have studied, isn't it? In muscle cells we have studied, in certain algae we have studied, isn't it? Sorry, in fungus we have studied. Okay, xenocytic condition, okay, means more than one nucleus per cell. Is it clear? So and there are certain cells which are not having any nucleus. Do you remember RBCs? RBCs are examples for cells which are not having nucleus. And sieve tubes of plants, do you remember, of phloem? So these are examples of cells without any nucleus. And when you see the matrix or the nucleoplasm, it contains what? Two main structures, that are nucleolus and the chromatin. Nucleolus or nucleoli are spherical structures which are not bounded by any membrane, and that is the site of rRNA synthesis. RNA, have you heard? DNA and RNA. RNA are of different types. Okay, so rRNA, that forms the structural part of ribosomes, they are actively synthesized in where? In the nucleolus. And chromatin is what actually is the thread-like structures that condense to form the chromosomes during or just before the cell division. Clear? So this is the structure of nucleus. So when the nucleus structure is asked, what all you have to write? Nuclear membrane having two layers, outer and inner, perinuclear space, nuclear pores, then matrix known as nucleoplasm consisting of what? Nucleolus and chromatin material. Okay, so the chromatin materials will get condensed to form chromosomes just before the cell get divided. Okay, so if you see the structure of chromosomes, okay, they or chromatin material, they chemically they contain what? DNA, some proteins, some non-histone proteins, okay, then some RNA. So these are the major content of what? Each chromatin material or chromosome. Okay, and chromosome will be formed from what? From the chromatin network itself by its condensation. They condense to form chromosomes. Okay, and if you see the human cell, there are each cell is having around 2 meter long, okay, DNA. Just imagine 2 meter, that much long DNA is condensed, okay, in each chromosomes. Means that 2 meter long DNA is condensed in 46 chromosomes of human cell. Okay, and this we will study in the next year, how this DNA is packed inside the chromosomes. Okay, now if you see the structure of a chromosome, okay, you can see that a constricted area, means a narrow area will be there. That narrow area is known as the centrosome. What it is? Centrosome. Okay, primary constriction or the centrosome, and on its side you can see a disc shaped structure, and that is known as the kinetochore. Okay, so you can see kinetochore is present on the side of the centrosome. Okay, what is the other name for centrosome? Primary constriction. Clear? And where this centrosome is placed, on that basis chromosomes are divided into 4 types. Okay, that is, first is the metacentric. Metacentric means the this kind of chromosomes are having its primary constriction or centrosome in the middle, so that they are having 2 equal arms. Okay, the chromosomes will have 2 equal arms, and the metacentric chromosomes are having centromere near to one end of the chromosome, so that what will happen, one arm of the chromosome will be larger and the other will be shorter. Okay, then another type is what, acrocentric. Acrocentric means here the centromere will be situated very close to the end of the chromosome. Okay, so that one arm will be extremely short and one will be extremely long. Okay, and telocentric means what, here the chromosome is having its centromere at its end, okay, terminal region. So on that basis chromosomes are what, of different types. 4 different types of chromosomes are there. What are they? Metacentric, okay, submetacentric, acrocentric and telocentric. Okay, so these are what, different chromosomes. So chromosomes are what? They are actually they are made up of what? DNA, proteins, histone proteins and non-histone proteins, and RNA. Okay, and depends upon they are formed from chromatin material by the condensation. Is it clear? So depends upon the position of the centromere, thereof chromosomes can be of 4 different types. Okay, and then some chromosomes, they are having a non-staining secondary constriction. Okay, means in addition to the centromere they will have one more constriction, and they gives the appearance of small fragment, and that is known as satellite. Okay, you can see here satellite, that will be giving the appearance of a satellite, and that kind of chromosomes are known as satellite chromosomes. Okay, so in addition to this, the cell may also contains various microbodies. Okay, they are what, actually membrane bound minute vesicles, means some vesicles which are bounded by membranes, and they may contain what, enzymes. Okay, which can be found in plants and animals. Okay, so now I think you got an idea about a cell, isn't it? Cell, if you see the overall structure of a cell, they will have cell membrane, cytoplasm and various organelles, and we have studied what about the various organelles present in the cells. So this is the major information about the cell. Okay, so you please draw the diagrams whichever it is given in this, and the main points you please write in your copy. Thank you.